Breaking the Cancer Shield. A new strategy to empower natural killer cells researchers have unveiled a promising new method to enhance the body's innate ability to fight cancer. By targeting specific proteins, scientists can supercharge natural killer NK cells, enabling them to penetrate the protective barriers that tumors build to evade the immune system. The barrier problem. Why NK cells often fail natural killer cells are the immune system's first responders. Their primary job is to identify and eliminate abnormal cells, including cancer. However, tumors are not passive targets. They often create a protective shield, a complex microenvironment that prevents NK cells from reaching them or suppresses their activity once they arrive. This biological defense mechanism allows the cancer to grow unchecked, even when the patient's immune system is theoretically capable of fighting it. A breakthrough in cell activation, a research team from McGill University's Rosalind and AMP. Morris Goodman Cancer Institute has discovered a way to bypass these defenses. By using small molecule drugs to block two specific proteins, the researchers found they could significantly boost the effectiveness of NK cells. In preclinical testing, this approach demonstrated remarkable results. Broad efficacy. The method successfully destroyed human cancer cells across various difficult-to-treat types, including leukemia, glioblastoma, kidney cancer, and triple-negative breast cancer. Tumor suppression. In animal models, the treatment effectively slowed the growth of tumors. Why this method is different. Safety and speed. Current immunotherapies often rely on genetic editing, which involves permanently altering a patient's cells. While effective, this carries risks of unintended genetic mutations and side effects. The new strategy offers three distinct advantages over traditional cell-based therapies: reversibility and safety. Instead of permanent genetic changes, this method uses small molecule drugs to temporarily enhance NK cell activity. This makes the treatment more controllable and potentially safer. Off-the-shelf availability. Most current immunotherapies require autologous treatment cells must be harvested from the patient, modified, and reinfused, a process that can take weeks. This new method uses NK cells derived from umbilical cord blood donations. These cells can be pre-prepared, stored, and administered to patients immediately. Scalability. Because the cells are not customized for each individual, the process is significantly faster and more affordable, making it easier to deploy in a clinical setting. The road ahead. While the preclinical results are highly encouraging, the transition to human patients is the next critical hurdle. The research team intends to prioritize acute myeloid leukemia, AML, a highly aggressive blood cancer, for upcoming clinical trials. The project is currently moving toward the regulatory approval phase and is seeking the necessary funding to begin human testing. This approach represents a shift toward off-the-shelf immunotherapy, potentially transforming cancer treatment from a slow, bespoke process into a rapid, scalable medical intervention. Conclusion. By combining pre-prepared donor cells with temporary chemical enhancement, scientists are creating a more accessible and potent way to break through cancer's defenses. If successful in clinical trials, this could provide a vital lifeline for patients who have exhausted standard treatment options.